Today's discussion is going to be on Chapter 4, Biology of the Cell. Chapter 3 objectives are to be able to recognize common features and general functions of cells, understand chemical structure and functions of plasma membranes, describe active and passive processes of membrane transport, describe cell communication, identify cellular structure and functions of each, and describe the structure and function of the nucleus, ribosomes, and cell division. Cytology is the study of cells, which are the fundamental units of life. Microscopy is the technique using microscopes to observe small-scale structures. There are different types of microscopes. Light microscopes uses visible light and glass lenses and produces 2D images of stained specimens. Electron microscopes employs electron beams and visualizes specimens stained by heavy metals. This includes transmission electron microscope, or TEM, which passes electrons through thin-cut sections of specimens. This visualizes internal structures in detail. Then there's scanning electron microscopes. This creates a detailed 3D image of a specimen's surface. Cells vary in size from microscopic to visible to the naked eye. Shapes include irregular, disc-shaped, cube-shaped, columnar, spherical, and cylindrical. Understanding cell size and shape is crucial for comprehending their function and behaviors. Cellular components include the plasma membrane, which is also known as the cell membrane. This forms the outer barrier separating internal cell contents from interstitial fluid. The nucleus is the largest structure enclosed by a nuclear envelope. It contains genetic material known as DNA. Cytoplasm, all cellular components between the plasma membrane and the nucleus. This comprises cytosol, organelles, and inclusions. Cytosol is syrup-like fluid with high water content. It contains dissolved macromolecules like carbohydrates, lipids, proteins, and small molecules. Organelles are complex structures within cells with unique shapes and functions, divided into membrane-bound and non-membrane-bound organelles. Cell inclusions are various nutrients or pigments within the cell without organelle activity. Examples include glycogen, lipids, and pigments like melanin. The general cell functions include maintaining cell integrity and shape, obtaining nutrients and forming chemical building blocks, disposing of waste, and some cells can undergo cell division to reproduce. Next, we're going to talk about the chemical structure of the plasma membrane. The plasma membrane includes lipids such as phospholipids, cholesterol, and glycolipids. Phospholipids are the predominant lipid type. They're structured like a balloon with a polar hydrophilic head and two nonpolar hydrophobic tails. They are arranged in parallel layers to form the phospholipid bilayer, defining the membrane structure. This ensures the separation of cytosol, which is on the inside of the cell, and interstitial fluid, which is on the outside of the cell. Cholesterol are a 4-ring lipid molecule. They are dispersed within the inner hydrophobic region of the phospholipid. This strengthens the membrane and assists in temperature regulation. Glycolipids are lipids with attached carbohydrate groups. Each group is linked to a phospholipid molecule on the outer layer of the plasma membrane. Membrane proteins are broken into structural types and functional types. Structural types include integral proteins, which are embedded within and extend completely across the phospholipid bilayer. Many are glycoproteins with attached carbohydrate groups. Also peripheral proteins, which are not embedded within the lipid bilayer. They are loosely attached to the external or internal surfaces of the membrane and often anchored to integral proteins. Functional categories include transport proteins, which regulate substance movement across the plasma membrane. There are various types, including channels, carrier proteins, pumps, some porters, and antiporters. Cell receptors bind specific molecules called ligands. Ligands are molecules that bind to macromolecules. Identity markers communicate cell belongingness. Enzymes catalyze chemical reactions either internally or externally. Anchoring sites secure the cytoskeleton to the plasma membrane. Cell adhesion proteins facilitate cell-to-cell attachments, forming membrane junctions with various functions. The primary functions of plasma membranes include physical barrier, which separates the cell from its external environment, acting as a protective boundary. Also selective permeability. This regulates the movement of materials in and out of the cell, allowing only specific substances to pass through. Establishment of electrochemical gradient. This creates and maintains differences in ion concentrations across the membrane, which is essential for cellular functions such as signaling and metabolism. Cellular communication. This facilitates interactions and signaling between cells, allowing for coordination of various cellular activities. Membrane transport can be broken into passive processes and active processes. Passive processes do not require cellular energy, and they include diffusion, facilitated diffusion, and osmosis. Active processes require cellular energy, usually in the form of ATP. These include active transport, endocytosis, and exocytosis. Passive processes, again, these processes do not require the expenditure of cellular energy. Instead, they rely on the kinetic energy of ions and molecules to move down their concentration gradients. Diffusion involves the movement of solutes from an area of high concentration to an area of low concentration. This occurs until equilibrium is reached, where there is equal distribution of solutes throughout the system. Osmosis specifically refers to the movement of water molecules across a semi-permeable membrane. Water moves from an area of higher concentration with a lower solute concentration to an area of lower water concentration that includes a higher solute concentration. This continues until equilibrium is established, balancing the concentration of solutes on both sides of the membrane. Diffusion is the movement of ions or molecules down their concentration gradient, driven by kinetic energy, which causes them to move randomly and constantly. The kinetic energy increases with temperature, leading to faster molecule motion. Characteristics of diffusion include dependence on a concentration gradient. Diffusion occurs from regions of higher concentration to regions of lower concentration following the concentration gradient. Also, spreading out of substances. The random motion of ions or molecules leads to their spreading out or diffusion from areas of high concentration to areas of low concentration. Attainment of equilibrium. Diffusion continues until equilibrium is reached, where there is an equal distribution of ions and molecules throughout the system. At equilibrium, there is no net movement of ions or molecules, as the rate of diffusion in one direction equals the rate in the opposite direction. Simple diffusion are when molecules that are small and nonpolar move unassisted into or out of a cell down their concentration gradient. These molecules move freely across the plasma membrane without the need for assistance from membrane proteins. Their small size and nonpolar nature allow them to pass through the lipid bilayer. Facilitated diffusion are small molecule solutes, such as uncharged ions or polar molecules, that are transported into or out of a cell down their concentration gradient with the assistance of plasma membrane proteins. This involves the movement of solutes through specific protein channels or carrier proteins embedded in the plasma membrane. Channel-mediated diffusion are when small ions move across the membrane through water-filled protein channels. Channels can be either leak channels, which are continuously open, or gated channels, which open in response to a specific stimuli. Carrier-mediated diffusion is when polar molecules larger than ions are transported by carrier proteins. This involves three main steps: binding of the polar molecule to the carrier protein, conformational change of the carrier protein to transport the molecule across the membrane, and the release of the molecule on the other side of the membrane. Osmosis is the passive movement of water through a semi-permeable membrane. Osmosis occurs in response to differences in water concentration on either side of the membrane. While water molecules move from an area of lower solute concentration to an area of higher solute concentration until equilibrium is reached. This process does not require energy. Osmotic pressure is the pressure exerted by the movement of water across a semi-permeable membrane due to the difference in water concentration. The magnitude of osmotic pressure increases with the steepness of the concentration gradient, leading to a higher movement of water molecules. Tonicity is the ability of a solution to change the volume or pressure of a cell by osmosis. Isotonic solution is a solution with the same relative concentration of solutes and water as the cell's cytosol. In an isotonic solution, there is no net movement of water across the cell membrane, resulting in the cell maintaining its normal volume and shape. Hypotonic solution is a solution with a lower concentration of solutes and a higher concentration of water compared to the cell's cytosol. In a hypotonic solution, water moves into the cell, causing it to swell and potentially burst. Hypertonic solution is a solution with a higher concentration of solutes and a lower concentration of water compared to the cell cytosol. In a hypertonic solution, water moves out of the cell, causing it to shrink or shrivel.
Here's a cell on your screen. We have already seen that the various organelles inside it are compactly packed inside this delicate membrane called the cell membrane or the plasma membrane. We've even seen that the cell membrane makes use of diffusion process to transport gas and water molecules in and out of the cell. However, diffusion is not the only process employed. There are a few more on the list. Let's have a look at these methods in this video. Tell me one thing, how do we transport water from a lower height to a higher height? We need a pump. Because this does not happen on its own. A force or some energy is required to pull the water upwards. Similarly, in a cell, some molecules have to be carried inside or moved outside the cell. This requires energy to be spent. The energy used is in the form of ATP molecules. This is called as active transport. That is, active transport requires expenditure of energy. The transportation is carried out by protein molecules present on the cell membrane. On the other hand, diffusion does not require energy. Hence, it's a type of passive transport. So, movement of molecules without spending energy, as in case of diffusion, is passive transport. So, Similarly, movement of molecules across the membrane with the expense of energy is active transport. Now let's get to know one more interesting concept named osmosis. Have we ever come across this term osmosis? Well, quite often. But what exactly do we mean by this? Osmosis is defined as the movement of water from higher water concentration to lower water concentration through a semi-permeable membrane. So let's say we have this container which is partitioned by this semi-permeable membrane. Now we add sugar solutions at both these sides. However, the concentration of each is different. Here we add concentrated sugar solution, while here dilute sugar solution. That means the concentration of water in this solution is obviously more compared to this. So after some time, we see water molecules moving from this side of the partition to this side. And how long will this movement take place? It will occur till the concentration of water is balanced, that is, equalized on both the sides. This is nothing but osmosis. Let's learn some more interesting concepts about osmosis and cell transport in the next video.
Active processes are cellular processes that require the expenditure of cellular energy, usually in the form of ATP, to move substances against their concentration gradient or to transport large molecules or particles across the cell membrane. In active transport, cellular energy is used to move a solute, which is an ion or molecule, up its concentration gradient from an area of lower concentration to an area of higher concentration. This process requires the use of specific transport proteins embedded in the cell membrane. Vesicular transport is when cellular energy can also be used for vesicular transport, which involves the movement of large molecules or particles into or out of a cell using a vesicle. This process includes endocytosis, which is the uptake of materials into the cell, or phagocytosis, which is cellular eating, or pinocytosis, which is cellular drinking. Exocytosis is the release of materials from the cell. Primary active transport directly utilizes energy derived from the breakdown of ATP. Energy released during ATP hydrolysis powers the movement of molecules against their concentration gradient. Sodium-potassium pump is an exchange pump that moves sodium ions out of the cell and potassium ions into the cell against their concentration gradient. This is crucial for maintaining the cell's electrochemical gradient, vital for nerve impulse transmission and muscle contraction. Secondary active transport is when energy stored in the electrochemical gradient of one molecule, usually sodium ions, drives the transport of another molecule against the concentration gradient. This doesn't directly require ATP hydrolysis, but depends on the energy provided by the ion gradient established by the primary active transport. Exocytosis, which is vesicular transport, is a process where large molecules or substances are secreted from the cell. This involves fusion of vesicles into the plasma membrane, releasing contents into the extracellular space. It is essential for secretion of hormones, neurotransmitters, and digestive enzymes. Endocytosis is cellular uptake of materials from the external environment into the cell. It is broken down into phagocytosis, which is cellular eating, where cells engulf large particles or pathogens by extending around them. It is primarily carried out by specialized immune cells like macrophages and neutrophils. Pinocytosis is cellular drinking. It involves internalization of small droplets of extracellular fluid containing dissolved solutes. Small regions of the plasma membrane invaginate to form vesicles. which then pinch off and enter the cell interior. Receptor-mediated endocytosis is a highly specialized form of endocytosis where binding of specific molecule receptors on cell surfaces. This enables selective internalization of substances from the cellular environment by recognizing and binding to ligands. Membrane potential refers to the electrical charge difference across the plasma membrane of a cell, representing potential energy. Resting membrane potential is when membrane potential of a cell is at rest and is crucial for normal function, especially in muscle and nerve cells. Establishing a resting membrane potential starts with an unequal distribution of ions. Cells have an uneven distribution of ions and charged molecules across the plasma membrane. Cytosol contains more potassium ions than the surrounding interstitial fluid, while sodium ions are more abundant outside the cell. Negatively charged protein molecules in the cytosol also contribute to the imbalance. Plasma membranes have more positive charges on the outside and more negative charges on the inside, creating a relative negative charge inside the cell compared to outside. This is measurable by electrodes. The resting membrane potential varies depending on cell type. Potassium is crucial for establishing resting membrane potential. Potassium ions exit the cell through leak channels, moving from the cytosol to the interstitial fluid, leaving the inside of the cell more negatively charged. Sodium ions enter the cell through leak channels driven by both chemical and electrical gradients, moving from the interstitial fluid into the cytosol. Maintaining a resting membrane potential using the sodium-potassium pumps is vital for maintaining concentration gradients of potassium and sodium established by diffusion. They actively transport sodium out of the cell and potassium into the cell, counteracting their passive movement. This process consumes cellular energy in the form of ATP.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss membrane potential. Membrane potential refers to the difference in electrical charge between the inside and the outside of a neuron. This is the plasma or cell membrane of the neuron. It separates the inside of the cell from the outside environment. We'll say this is the inside, and this is the outside of the neuron. The difference in electrical charge develops due to the grouping of ions on the inside and the outside of the membrane. Ions are atoms that have either lost or gained electrons and thus have a positive or negative charge. There are several ions that play an important role in the membrane potential of neurons. There are positively charged sodium ions, represented by these blue circles, and negatively charged chloride ions, represented by these green circles. When a neuron is at rest, the sodium ions and chloride ions are more prevalent outside of the cell. There are also positively charged potassium ions, represented by these yellow circles, and various negatively charged ions, often referred to as organic anions, represented by these gray circles. Anion is simply a term for a negatively charged ion. When a neuron is at rest, the potassium ions and organic anions are more prevalent inside the cell. At rest, the inside of the neuron is more negatively charged than the outside, causing the resting membrane potential of an average neuron to be around negative 70 millivolts. One way this potential is maintained is through a mechanism known as the sodium-potassium pump. This is a transport protein that uses energy to constantly pump 3 sodium ions out of the cell, while at the same time pumping 2 potassium ions into the cell. Because there are more positive ions being pumped out than in, it helps to keep the membrane potential negative. Unlike other ions, potassium tends to move fairly easily across the cell membrane through ion channels, which are membrane-spanning proteins that allow ions to pass through. Potassium will pass out of the neuron until it reaches the point where it is at an equilibrium, when forces like diffusion aren't pushing it in one direction or the other. At this point, the membrane potential of the neuron is around negative 70 millivolts, which is known as the resting membrane potential.
Next, we're going to discuss cellular communication via the plasma membrane. There is direct cell contact, which is essential for various cellular functions, particularly in the immune system. This facilitates communication between immune cells and other cells, aiding in the identification and elimination of foreign or unhealthy cells. It is crucial for cellular regrowth and repair processes, especially after injury. Ligand receptor signaling is the most communication between cells occurs through ligands, which are molecules that bind to specific macromolecules, also known as receptors. There are types of ligands. There are neurotransmitters, which are released from nerve cells, and hormones, which are released from endocrine cells. Target cells possess receptors that can bind to these ligands, facilitating cellular communication. There are types of receptors. There's channel-leaked receptors, or chemically gated channels. They permit the passage of ions into or out of the cell upon ligand bonding. There are enzyme receptors, which act as protein kinase enzymes becoming activated upon ligand binding. Activated enzymes directly add phosphate groups to other enzymes within the cell. There are G protein coupled receptors. They utilize G proteins as intermediates in the signaling process. Ligand binding activates G proteins. The activated G proteins then activate protein kinase enzymes. These enzymes initiate various cellular responses within the target cells. Cellular communication via the plasma membrane involves both direct cell contact and ligand receptor signaling. Direct contact is crucial for immune function and cellular repair, while ligand receptors interactions facilitate a wide range of cellular responses through different types of receptors. Here we're going to talk about membrane-bound organelles. First, we have endoplasmic reticulum, or ER, which is an extensive network of interconnected membranes within the continuous lumen. There's rough endoplasmic reticulum, which has ribosomes attached and produces proteins for secretion, membrane incorporation, and lysosomal use. Smooth endoplasmic reticulum synthesizes lipids, metabolizes carbohydrates, and detoxifies drugs. The Golgi apparatus is composed of flattened membrane-bound sacs. It modifies, packages, and sorts proteins synthesized by the rough endoplasmic reticulum for secretion or use within the cell. Lysosomes are membrane-enclosed spherical sacs containing digestive enzymes in an acidic environment. They are involved in intracellular digestion and the recycling of cellular components. Peroxisomes are membrane-bound organelles containing various enzymes. They perform diverse metabolic functions, including the breakdown of fatty acids and detoxification of harmful substances. The endomembrane system is an interconnected system of membrane-bound organelles, including the endoplasmic reticulum, Golgi apparatus, vesicles, lysosomes, and peroxisomes. The mitochondria is an oblong-shaped organelle with a double membrane and inner folds called cristae. It contains their own DNA and synthesize ATP through aerobic cellular respiration, utilizing glucose and other fuel molecules for energy production. Here we're going to discuss non-membrane-bound organelles. Ribosomes are composed of protein and RNA. They exist as either free-floating or bound to endoplasmic reticulum membrane. They synthesize proteins based on instructions from messenger RNA. Centrosomes contain a pair of centrioles surrounded by pericentriolar material. They play a key role in organizing microtubules during cell division, forming the mitotic spindle. Proteasomes are barrel-shaped protein complexes responsible for protein degradation. They break down damaged or unneeded proteins into smaller peptides for recycling or disposal. The cytoskeleton provides structural support and organization within the cell and is composed of 3 types of protein filaments. Microfilaments, which is actin filaments, which is involved in cell movement and support. Microtubules form tracks for intracellular transport and help maintain cell shape. And intermediate filaments, which provide mechanical strength and stability to the cell. Structures of the cell's external surface include cilia, which are small, hair-like projections extending from exposed surfaces of some cells. They are found in large numbers on epithelial surfaces, such as the respiratory tract, and assist in moving mucus and other substances across the cell surface. Flagella are longer and wider than cilia. They propel the entire cells, such as sperm cells, through their environment. They are found singly or in small numbers of cells. Microvilli are microscopic membrane extensions of the plasma membrane. They are densely packed together on the surface of cells, particularly in the intestines and kidneys. This helps to increase the surface area of the plasma membrane for more efficient absorption and secretion. Membrane junctions include tight junctions, which form a continuous seal around the apical end of epithelial cells. They prevent leakage of extracellular fluid and regulate passage of ions and molecules between cells. Desmosomes are anchoring junctions composed of proteins that bind neighboring cells together. They provide mechanical strength and stability to the tissues subjected to mechanical stress, such as the skin and cardiac muscle. Gap junctions consist of clusters of integral membrane proteins. They form small fluid-filled channels that allow direct communication and exchange of ions and small molecules between adjacent cells. They are important for coordinating cell activities in tissues like cardiac muscle and smooth muscle. Next, we're going to discuss the structures of the nucleus. There is the nuclear envelope, which is a double membrane that surrounds the nucleus. It separates the nucleoplasm, which is the fluid within the nucleus, from the cytoplasm. It contains nuclear pores for exchange of materials between the nucleus and the cytoplasm. It is associated with rough endoplasmic reticulum on its exterior. The nucleolus is a non-membrane bound structure located within the nucleus. It is composed of proteins and RNA and is responsible for synthesizing ribosomal RNA and assembling ribosomal subunits. It appears as a dark, staining spherical body. DNA, or deoxyribonucleic acid, is a double helix structure consisting of 2 strands of nucleotides. Nucleotides composed of 5 carbon sugar, deoxyribose, a phosphate group, and 1 of 4 nitrogenous bases, which include adenine, cytosine, guanine, and thymine. Hydrogen bonds hold complementary base pairs together, A with T and C with G. Chromosomes are made up of DNA tightly coiled around histone proteins. Chromatin is a complex of DNA, histone proteins, and other proteins found within the nucleus. It exists in a loosely packed form during interphase, which is non-dividing phase, of the cell cycle. It condenses into visible chromosomes during cell division, known as mitosis or meiosis. It contains the genetic information necessary for cell function and inheritance. Transcription is the process of synthesizing RNA from a DNA template. A DNA template, ribonucleotides A, U, C, and G, and RNA polymerase enzyme are required structures. The first step is initiation, where RNA polymerase binds to the promoter region of the DNA. Then there is elongation, where RNA polymerase moves along the DNA template, synthesizing RNA by adding complementary ribonucleotides. The termination phase, RNA polymerase reaches the terminator sequence, causing RNA synthesis to stop. Modifications to messenger RNA include splicing, which is the removal of introns or non-coding regions, and joining of exons, which are the coding regions, to form mature messenger RNA. Also capping, which is an addition of a 5 cap to the 5 end of the messenger RNA. The poly-A tail is the addition of polyadenylated tail, or poly-A tail, to the 3 end of the messenger RNA. Translation is synthesizing proteins from messenger RNA. The required structures include ribosomes, messenger RNA, template, transfer RNA, and free amino acids. The steps involved are initiation, elongation, and termination. DNA contains the genetic information necessary for directing the synthesis of proteins. Through transcription and translation processes, DNA controls the synthesis of proteins that carry out various cellular functions. The regulation of gene expression allows cells to respond to internal and external cues and maintain homeostasis.
In this video, we're going to talk about transcription and translation. And here is just a basic overview of these two processes. Transcription is the process by which DNA gets converted into mRNA, also known as messenger RNA. And translation is the process of converting the information stored in messenger RNA and using it to build protein. Now, if you recall, which organ now is used to make proteins? What would you say? The answer is the ribosome. The ribosome is the site at which proteins are manufactured in the cell. So we're going to talk about that shortly. But let's go over transcription first before we talk about translation. Transcription occurs in 3 steps: initiation, elongation, and termination. Now, during initiation, RNA polymerase binds to the promoter region of DNA. Now, what is the promoter region? The promoter region is basically a short sequence of DNA. In eukaryotic DNA, it's TA, also known as the Theta box. Now, this sequence is located 25 nucleotides upstream of the site where transcription begins. The next thing that RNA polymerase does is it causes the 2 DNA strands to separate. And during elongation, it begins to add nucleotides to the growing mRNA strand that we see here. Thus, RNA polymerase synthesizes mRNA starting from the 5' end going to the 3' end. However, RNA polymerase, it reads the DNA strand in the 3 to the 5' direction. Now, there's 2 strands that you need to be familiar with. The first one, the one that's used to synthesize mRNA, is called the template strand, or the antisense strand. So that's the one in which RNA polymerase is active upon. The other one, which is not used, this is called the the non-template strand, also known as the sense strand. Some textbooks will call it the coding strand because its sequence matches up with RNA, except the fact that uracil is found in RNA, but thymine is found in DNA. Now, during the last step of transcription, which is the termination step, the RNA polymerase molecule, the mRNA strand, they all separate from the DNA template strand. Now, in this step, the poly A polymerase enzyme, it caps the free end of the mRNA strand, and this is known as the poly A tail. Now, also during the beginning of transcription, the 5' end is also capped, and the reason for this is to protect the mRNA strand from being degraded by certain enzymes. At the end of the termination step during transcription, DNA has been used to create a pre-messenger RNA strand. Now, this particular strand has something known as introns and exons. Introns are basically longer sequences of nucleotides that do not code for anything. So these, they must be removed in a process known as RNA splicing. Now the exons, those are shorter sequences of nucleotides, and they're going to be used to synthesize proteins. And so the exons remain, but the introns, they must be removed. And so now we have a completed messenger RNA strand. Now let's work on a practice problem. Let's say if you're given a sequence of nucleotides on a DNA strand, and you're asked to write the corresponding sequence on an mRNA strand. What would it be? Feel free to pause the video and try it. So if we're reading the DNA strand from the 3 to 5 direction, we're going to have to write the corresponding mRNA sequence in the 5 to 3 direction. So what letter corresponds to G? It's important to know that G always corresponds to C, and vice versa. C corresponds to G. Now, what letter corresponds to A? A usually corresponds to T, but there's no T in RNA. Instead, A is going to correspond to U for uracil, but T in DNA corresponds to A in RNA. And so if we continue, everything else is going to be U, C, A, U, A, U, G, C. And so this is the mRNA strand that corresponds to the nucleotide sequence in DNA listed above. Now let's talk about translation, which is the process of taking the information stored on an mRNA strand and using it to construct a protein. So once the mRNA strand is synthesized in the nucleus, it leaves the nucleus and enters the cytosol, where it interacts with a free ribosome or one that is attached to the rough ER. Now within the ribosome, it's going to interact with a tRNA molecule or a transfer RNA molecule. Now let's focus on the sequence of nucleotides on the mRNA strand. Notice that they're separated in sets of 3. Each set of 3 nucleotides represents a codon, which matches up with another 3 nucleotides on a tRNA molecule known as an anticodon. And each codon matches up with a specific amino acid. And so that's how the information stored in the mRNA strand can be used to construct a specific protein. And we're going to talk more about this later in this video. Translation, like transcription,
The cell is the fundamental unit of life, comprising various organelles, membranes, and cytoplasm enclosed within a plasma membrane. The cell cycle consists of interphase and the mitotic phase, ensuring proper growth, DNA replication, and division. Interphase is broken down into G1 phase or GAP1. The cell grows in size. It performs normal metabolic activities. It synthesizes proteins necessary for DNA replication, and the duration varies depending on the cell type. Next is S phase or synthesis. DNA replication occurs, producing two identical sets of chromosomes. Each chromosome consists of two sister chromatids joined at the centromere. Centrosomes is also duplicated. G2 phase or GAP2, the cell continues to grow. It produces proteins and organelles and prepares for mitosis by synthesizing components of the mitotic spindle. Final checks to ensure DNA replication is done correctly. In the mitotic phase or M phase, divided into mitosis, nuclear division, and cytokinase, cytoplasmic division. In mitosis, you have prophase, where the chromatin condenses into visible chromosomes. The nuclear envelope begins to break down. Spindle fibers emerge from the centrosomes. Centrosomes move to the opposite pole of the cell. In prometaphase, the nuclear envelope fragments completely. Spindle fibers attach to kinetochores on the centromeres of the chromosomes. Chromosomes begin to move toward the cell center. In metaphase, chromosomes align at the metaphase plate, which is the center of the cell. Spindle fibers are fully attached to kinetochores of each chromosome. Anaphase, sister chromatids separate and are pulled to opposite poles by spindle fibers. The cell elongates as spindle fibers push poles apart. Telophase, the chromatids reach opposite poles and decondense into chromatin. New nuclear envelopes form around each set of chromosomes. Spindle fibers disassemble. Cytokinase is the division of cytoplasm to form two separate daughter cells. In animal cells, a cleavage furrow forms, pinching the cell into two. In plant cells, a cell plate forms, eventually becoming the cell wall. The cell cycle is regulated by a complex set of signaling pathways involving cyclin-dependent kinase, and checkpoint proteins to ensure accurate DNA replication and division. Dysregulation of the cell cycle can lead to uncontrolled cell growth and cancer.
Hi, it's Mr. Anderson, and in this video I'm going to talk about cells. Life is made of cells. It's the basic unit of structure and function. And also, all cells we have must come from pre-existing cells. And so how does that work? It's through a process called cell division. This is going to occur in all cells, both prokaryotic like bacteria and eukaryotic like you. And through cell division, one cell can become 2, which can quickly become 4, and then 8, and then 16, and so we get exponential growth in cells. And to give you an idea how quickly this goes, you started life as one fertilized egg or one zygote. But those were quickly dividing over time, and it eventually created an adult that's going to have around 10 trillion cells inside your body. And they all came from that original cell. Now, to even increase that number quite a bit, just look in your gut, and we're going to find that the bacteria living in your digestive tract are going to outnumber the cells in your whole body 10 to 1. There are going to be 100 trillion bacteria just living in your gut, and they're going to be outnumbered about 10 to 1 by viruses living in your gut. So we're going to have quadrillion viruses living in your gut. Now, viruses aren't technically cells, but everything else I've described here is going to be reproducing, or it's going to be created through a process called cell division. And there are really three types of cell division. In prokaryotic cells, or in bacteria, they're simply going to divide through a process called binary fission. That's where one cell, one bacteria cell, is going to make two cells that are exact copies of the original cell. In eukaryotic cells, we have something similar to this. We're going to have cell division, or we sometimes just refer to it as mitosis. Technically, mitosis is going to be division of the nuclei, and then we're going to have cytokinesis, which is division of the rest of the cell. But we generally just say mitosis is when one eukaryotic cell makes two cells that are exact copies of that cell. It's a lot like binary fission. And then the third type of cell division is going to be meiosis. Meiosis is going to be when we create sex cells, or when we create gametes. Mitosis and binary fission ensure that we have exact duplicates of that original cell. Meiosis is totally different. Meiosis is going to make cells that are different. They're going to have half the genetic information, and they're going to give us variability. And so in us, sex is important because it gives us variability in the next generation. In bacteria, mutation, and there are some other ways that they can share information is more important. So let's start by talking about bacteria. They reproduce through a process called binary fission. In this, we're going to have this adult cell, so we're going to have this parent cell. It's going to be made up of, you can see here, ribosomes. It's going to have a plasmid here, which is a little bit of extra DNA. But its genome, or all of its DNA, is going to be wrapped up into one loop. And so what do we want to do in binary fission? We want to take one cell and we want to make two cells that are identical to that. And so the process is pretty simple. What the bacteria cell starts doing is duplicating all of its DNA. So the first thing it'll do is DNA polymerase goes around the whole loop, and it's going to make an exact copy of all the DNA in the bacteria. One thing not shown in here is that we're also going to duplicate our plasmids, and we're also going to have, ribosomes increasing as we increase the number of DNA as well. What happens next is it's going to be pulled to the side of that cell, and really what's going to happen is the DNA will actually attach to the cell wall, and then eventually that cell wall is going to split in half, and we're going to have two bacteria cells. How are they different? They're not really different. They're exactly the same. The DNA in this cell and the DNA in this cell is going to be exactly the same. And when we divide in half, we're going to divide in half those ribosomes and the plasmids as well. And so again, bacteria don't do mitosis. They don't have sex. They simply reproduce through a binary fission. And this happens incredibly quickly. So in a typical E. coli, they can copy themselves, make two copies of one cell in 20 minutes, which is just crazy how fast that occurs. But again, we don't go through binary fission because we're eukaryotic cells. And before we talk about mitosis and meiosis, we should make sure you know the difference between what a gamete is and then a somatic cell. And so eukaryotic cells are going to be everything from the chimpanzee to the bees, to the flowers, to the mushroom, to the yeast. They all have a nuclei. And so there are two types of cells that we find inside eukaryotic organisms. First are going to be the somatic cells. Somatic cells are going to be the body cells in your body. And then we're going to have the gametes, which are going to be the sex cells. So in humans, the only cells inside our body that are going to be gametes are going to be sperm in males, and then the eggs or the ovum that are found inside females. How are they different? Remember, somatic cells, or the body cells, are going to be diploid in humans. They're going to have two copies of every gene and therefore two copies of every chromosome. Gametes are only going to have one. So sperm and egg each provide you with half the DNA that you get. So how do we duplicate a typical cell? Well, first we should say, when do we do that? Well, remember, when you go from a zygote to an adult, we're doubling the cells over and over and over again. And then we're constantly replacing the cells in our body. So as cells become worn out or damaged or removed, we have to replace those with other cells. And we do that through mitosis. Again, what is mitosis? It's technically the division of the nuclei, but then we're going to have division of the rest of the cell, which is called cytokinesis. So what do we start with? And we're going to make this real simple. This is a simple cell. It's a diploid cell. It's 2n equals 2. That means they have two chromosomes. These two chromosomes are going to be homologous. That means that they're the same length. The reason why they're different colors is you get one from one parent and one from another. And so in mitosis, let's look first, before we talk about the specifics of what we're going to get out of mitosis. We're going to start with one cell looks like this, and we're going to end up with two cells that look exactly like that first cell. So in mitosis, we want to duplicate the cell. And so let's watch that. What's the first thing that happens is we have to duplicate all of the DNA. and this occurs during interphase, what's going to happen is this chromosome right here is going to copy all of its DNA. And now we're going to have what are called sister chromatids. And so we have a chromosome on this side, a chromosome on this side, and they're exactly the same. So they're an exact duplicate of that original chromosome. And you can see that we're going to have that on the other homologous chromosome as well. What happens next, they're going to meet in the middle, they're going to line up. These lines represent a spindle which is going to attach to either side, each of those sister chromatids, and then they're going to be pulled apart. They're going to be pulled apart. And eventually, so this would be the mitosis when we divide the nuclei, and then the whole cell is going to split in half. We're going to divide that cytoplasm, and that's called cytokinesis. And so what do we end up with? We end up with two cells that are both diploid. Our original cell was diploid as well. So let's take a look at what this really looks like. We're looking right here at cells that are undergoing mitosis, and so they're growing on a growth medium. And if we just play the video, this is time lapse, and so it's speeding it up quite a bit. But what will happen if we look at like this cell right down here is that you can see it kind of come together, and the nuclei is dividing up the genetic information, and then the rest of the cell is going to divide. What do we end up with when we're done with that? We have two identical cells, and those are going to grow, and then they're eventually going to divide the nuclei, and they're going to grow, and they're going to grow, and they're going to grow. And cells, unless they're cancerous cells, are going to keep growing on here until they fill up this whole medium. But we need to talk about meiosis, which is the third type of cell division. What happens in meiosis? Meiosis is when you take one cell and we're going to make 4 cells out of it. Now let's look how this is a little different than in mitosis. In mitosis, remember, we took one cell and we made 2 cells. In meiosis, we're actually going to have another division. But there's something even different that we didn't see in mitosis. Remember, in mitosis, we want to make sure that each of those cells that we make are identical to that original cell. You can see here that the 4 cells that we create at the end are going to be totally different than that original cell. And they're going to have half the genetic information. Remember, I said the sperm and the egg have half the information that's found in the original cell. And so what's going to happen? Well, let's watch how this begins. First thing that happens is you're going to duplicate each of these chromosomes. And so we're going to make copies of the chromosomes, but then something odd happens. What happens that's odd, we're going to take a little bit, in this case we're going to take a little bit of this white chromosome here, and it's going to cross over with this red chromosome right here. So we're actually swapping chromosomes. And so that process is called crossing over, but what you're really doing is you're crossing over, or you're making a new chromosome. It's a combination of the chromosome you got from your mom and your dad. It's a chromosome that's never been built before. So those will eventually line up in the middle, and then they're going to divide, and then they're going to divide again. And so they'll divide in half, so this is meiosis 1, and then they'll divide in half again, and that's called meiosis 2. So what do we create when we're done? We're going to create daughter cells, but each of those daughter cells are going to be different. Each of these would create a new sperm or a new egg. Now it's going to be much more complicated in us. In humans, remember, we're not going to start with 2n equals 4, with 4 total chromosomes here. We're going to start with 2n equals 46, and so we're going to start with 46 chromosomes. So it's going to be really, really different when you get to the end. And so we can create literally billions and billions of different types of cells through meiosis. And so that's going to give us variation. So to summarize, what do we have? We have 3 types of cell division. We have binary fission. That's going to be found just in bacteria, where you take one bacteria, first copy the DNA, and then the rest of the cell is going to divide through cytokinesis. Each of these daughter cells is going to be identical to that first cell. If you're looking at eukaryotic cells, we've got mitosis. What happens in mitosis? We start with a diploid cell. So here the reds and the greens are each going to come from different parents. So we have 4 chromosomes. They're going to duplicate first during S phase of interphase. They'll line up in the middle, and they'll be pulled to the side. Now, if we look here, these 2 cells that are produced in mitosis are identical to that original cell. And that's what mitosis does. It makes exact copies. If we start with meiosis, meiosis is going to start with an original cell that looks very similar. But instead of lining up like this, where we separate the sister chromatids, they're going to line up with the homologous chromosomes wrapping around each other and swapping bits of that chromosome. They'll divide in half, and then they're going to divide in half again. And what we're going to create is gametes. Those are going to be sperm and egg that have half the genetic information and a new combination of that genetic information. So again, this will be fertilized by a different sperm, and we get back to a zygote. Where do we go from the zygote? We go right back here to mitosis, where we can copy that cell over and over and over and over again. And so that's cell division. It's found in all cells on our planet. It shows us how one cell can go to all the cells that we have today. A good question might be, where did that original cell come from? But that's for another podcast, and I hope that was helpful.
Aging is a natural and ongoing process that occurs throughout life, involving gradual changes at the molecular, cellular, and organismal level. These changes accumulate over time, leading to characteristic signs and symptoms associated with aging. Complex interactions are the mechanisms of aging that are complex and not fully understood, involving interactions between genetic, environmental, and lifestyle factors. Certain genes are associated with longevity and the regulation of the aging process. External factors such as diet, exercise, exposure to toxins, and stress levels can influence the rate and nature of aging. Aging results in a decline in metabolic functions, leading to decreased ability to maintain homeostasis. Cells become less efficient in repair and maintenance processes, contributing to the gradual decline in bodily functions. Cell death mechanisms can occur due to mechanical damage or exposure to harmful agents. External factors include injuries, physical stress, and exposure to toxins or harmful agents that can lead to cell death. Over time, cumulative damage from these external factors can accelerate the aging process. Apoptosis is programmed cell death. It is a controlled and necessary process for development and tissue turnover and maintaining cellular balance. Proper regulation of apoptosis is crucial. Dysregulation can lead to diseases such as cancer, which is reduced apoptosis, or neurodegenerative diseases, which is an increased apoptosis. Aging involves complex interplay of various factors, including genetic predispositions, environmental influences, and lifestyle choices. Aging affects cellular functions, leading to a decrease in the ability of cells to divide, repair, and maintain themselves. Over time, these cellular changes lead to alterations in tissues and organs, impacting overall physiology and health. Common physical changes, including wrinkling of skin, reduced muscle mass and strength, and decreased bone density, are normal physical changes. Aging can lead to slower metabolic rates, decreased immune function, and impaired cognitive abilities. So in conclusion, aging is a natural, complex, and multifaceted process influenced by a variety of internal and external factors. Understanding the mechanisms of aging can help in developing strategies to promote healthy aging and improve the quality of life in older adults.